last time I think I stopped in this, right? So what this slide is trying to say is that, alpha toxin produced by Clostridium perfringens, it can cause occlusion of the blood vessel. Okay. So how it causes occlusion of the blood vessel, Bandakiri, it causes, attachment of platelet with the leukocyte. Okay. So there will be so many attachment of so many platelet with so many leukocyte forming a mass. That mass can block the blood vessel, okay, of the affected part of the body by Clostridium perfringens. And there will be occlusion of the blood vessel, further decreasing the blood supply to the organ, causing necrosis or ischemic necrosis of the organ. I told you that there will be activation of platelet adhesion molecule GP2B3A. Okay, this GP2B3A will be activated, and there will be upregulation of this leukocyte receptor called as CD11B, CD18. So there is upregulation of this receptor and this receptor. And these two receptors will be bridged, will be connected by fibrinogen, okay, on the one hand. On the other hand, also, there will be upregulation of P-selectin of platelet, and also there will be, upregulation of another P-selectin glycoprotein ligand 1. So this P-selectin can bind to this, also some of the carbohydrate, okay, present on the leukocyte surface can bind to the platelet. In this way, there will be connection, there will be attachment of platelet with the leukocyte, which normally does not occur, forming a huge mass which can block the blood vessel and cause ischemic necrosis of the organ. Okay, that is what I am trying to say in this. Okay, did you understand? Buzio?
Yes, sir.
Okay, can I move on to the next slide?
Yes, sir.
Okay. So now let's talk about the clinical features of gas gangrene. Okay, increasing pain, tenderness, and edema of affected part. So that affected part, the patient has got, increasing pain. The pain goes on increasing, and also when you feel that part, the patient has got pain. That is called tenderness, and even there is swelling, what you call as edema. Necrotic black skin. Okay, the skin becomes black with large blisters. Okay, now you can see this patient has got some kind of trauma of this thigh, of right thigh and right hip. You can see the skin has become black, and you can see lots of blisters. Okay, so this blister, this large blister is called as bulla. If it is small in size, it is called blister. If it is very large in size, it is called as bulla. So these bullas are filled with fluid. Okay, it is fluid with necrotic fluid or the exudate. Okay, that is called bulla. Gas production by fermentation of glucose. Now our muscle, our tissue has got glucose. That glucose will be utilized by the anaerobic bacteria, that is Clostridium perfringens or Clostridium septicum, and there will be production of plenty of gas. So even gas will be produced just below the skin, in the muscles. Okay. Now, unlike other anaerobic infection, the discharge, okay, the discharge means that is the fluid. Okay, the fluid, it is not purulent. Okay, it is not abscess like yellow purulent, but the discharge is thin, watery. Okay. It is thin, watery discharge. It is profuse and serosanguinous. Serosanguinous means it looks like a serum mixed with blood. Okay, and that serosanguinous discharge or the fluid is because of lysis of neutrophils by the lecithinase. Now I told you that also the bacteria, it produces enzyme called as lecithinase. That lecithinase can cause lysis of neutrophil, and that produces that watery serosanguinous fluid, which looks something like this. Okay, so this kind of serosanguinous fluid might be present within this bulla or within this lesion of the patient. Okay, so systemic signs of toxemia and death due to septic shock. Okay, the patient can undergo septic shock. Okay, now what is septic shock? I must have to explain. I'll show you one figure below that will explain septic shock. So whenever there is any kind of some kind of foreign material or some kind of inflammation inside our body, there is SIRS. Okay, systemic inflammatory response syndrome. Okay, systemic inflammatory response syndrome is what our body, what our immune system does, or what our immune system responds in order, when there is some kind of foreign antigen or a bacteria infection inside the body. Okay, now when this SIRS or systemic inflammatory response becomes more severe, there is higher grade of infection. The infection has spread to many parts of the body, and the patient has got fever, hypotension, or altered sensorium. That is called as sepsis. Okay, now this sepsis will again become further worsened. That is called severe sepsis. There is sepsis, and along with sepsis there will be hypoperfusion. Okay, our body organs, the vital organ like the heart, brain, kidney, liver, they might get, less perfusion of the blood. Okay. So that is what you call as severe sepsis, at least one organ. Okay. This severe sepsis, if become more worse, it can go into septic shock. So septic shock means there is severe sepsis along with, hypotension. And even if you provide lots of fluid to the patient, even if you are providing fluid to the patient, that is not going to respond. The patient is not going to improve. Unresponsive to volume replacement. Okay, that is called as septic shock. So even this, Clostridium perfringens and gas gangrene, if not treated in time, if not action taken in time, if the patient is not taken any medical care, might undergo septic shock. And when the patient undergoes septic shock, the patient has to be admitted to the ICU, intensive care unit. Okay, and it is very difficult to save the life of the patient if the patient goes to septic shock. So this septic shock can be very serious complication of this gas gangrene. The part of the body which has been infected should be removed. This, this limb, this leg might have to be cut, might have to be amputated, okay, to save the life of the patient. Okay. So did you understand what is septic shock? There is severe sepsis along with hypotension. And even if you are providing lots of fluid to the body of the patient is not responding. Okay. Unresponsive to volume replacement, that is called as septic shock. It is the severe form of infection. Okay. Did you understand, all of you?
Yes, sir.
Hello. बुझ्यो?
Yes, sir.
Septic shock भनेको बुझ्यो?
Yes, sir.
बुझ्यो? यसको कुरा हुन्छ तिमीहरुको पछि हेमाटोपोइटिक सिस्टम सुरु भएको छ कि छैन तिमीहरुको? हेमाटोपोइटिक छैन है?
No, sir.
हेमाटोपोइटिक सिस्टममा सेप्टिक शकको बारेमा अझ राम्रोसँग पढाउँछ है। त प्याथोजेनेसिस अफ सेप्टिक शक हाम्रो माइक्रोबायोलोजीमा पनि प्याथोलोजीमा पनि पढाउँछ है। ओके, अहिले चाहिँ यति मात्र बुझ्यो कि सेप्टिक शक भनेको सिभियर फर्म अफ इन्फेक्सन हो जसमा पेशेन्टलाई फिभर, हाइपोटेन्सन हुन्छ र जति पेशेन्टलाई फ्लुइड चढाए पनि हैन, नर्मल सलाइनहरु फ्लुइड जति दिए पनि पेशेन्टलाई ठिक हुँदै हुँदैन हैन, पेशेन्ट रेस्पोन्ड गर्दै गर्दैन, एकदमै रेस्पोन्ड गर्न गाह्रो हुन्छ है। एन्ड द पेशेन्ट इज भेरी सिभियरली इल, द पेशेन्ट मस्ट हैव टु बी एडमिटेड टु आईसीयू। ओके, did you understand? Buzio?
Yes, sir. Yes, sir.
Can I move on to the next slide?
Yes, sir.
Okay. So again, to show you some of the picture of gas gangrene, just look at this picture. This patient has got some kind of, wound. Okay. So this wound has been sutured and still there is, gangrene of this, the closed part of the skin. Okay, there is blackish discoloration. This is something, necrosis has occurred here and there is gangrene. Okay, here you can see this again the case of gas gangrene. The skin has been already removed, peeled off here. Here also you can see. Okay, so the areas in our body which are very prone to gas gangrene, like they say commonly axilla, thigh, buttock, calf. Okay, these are the area which commonly suffer from gas gangrene. And if you closely look at the patient lesion having gas gangrene, okay, you'll find plenty of empty space, okay, having gas pocket, and that gas pocket area is surrounded by purplish black. Okay, this kind of purplish black lesion. Just below that, slightly below, you'll find olive green coloration. Okay, so it is not clear in this, but just below that black coloration you'll find olive green coloration, and again below that you find brick red color. Okay, and below that brick red color there is normal, okay, normal tissue or normal skin. Okay, so these are, different, parts of the gas gangrene affected. This is the most necrotic part. Okay, this is the slightly affected. This might be the least affected, and this is the normal. Okay, these are different kinds of, different parts you find in a gas gangrene lesion. Okay, did you understand?
Yes,
hello. Can I move on to the next slide?
Yes, sir.
Yes, sir.
Okay, so let's look at this patient. Okay, now this patient is suffering from gas gangrene or Clostridium perfringens infection of right thigh. Okay, the right entire thigh. Okay, now if this patient is not treated in time, the toxin produced in this part is going to spread throughout the whole body. The patient might die of septic shock. Okay, so the doctor, they are trying to remove this part. This whole right leg, okay, has to be cut and removed. That is called as the amputation. Okay, so before right leg amputation of the patient, gas gangrene, the right thigh is swollen, edematous, and you can see discolored, necrotic bulla, large blisters. Okay, and impressive crepitation. If you feel this, if you feel this with your hand, you'll feel something like there is crepitation. There is some kind of crackling sound, okay, if you feel, because of gas, okay. So since there is gas below the skin, normally we don't have gas below the skin. So when there is accumulation of gas below the skin because of gas gangrene, you'll feel some kind of crackling sound when you feel, that is called crepitation, okay, at this junction, and the patient is in shock, okay. So the treatment was done. The patient's whole right leg, including this what you call as the thigh leg, even the femur from the hip joint has been removed, okay. Of course, the patient is going to become handicapped. This patient cannot walk. The patient have to use the wheelchair, but at least the patient will survive, okay. This is called hemipelvectomy, okay. So hemipelvectomy means half of this, pelvis along with this leg has been totally removed to save the life of the patient, okay. So this patient is going to become handicapped, but the patient will live. The patient will survive, okay. So this is one of the treatment method for gas gangrene, okay. Did you understand?
Yes, sir. Yes, sir.
Can I move on to the next slide?
Yes, sir.
So now this is the histopathology. This is a muscle biopsy taken from a patient with gas gangrene. This is hematoxylin eosin stained, muscle slide, the muscle smear. So you can see this is the normal muscle, and can you see some empty space? So these all empty space is because of gas formation. There is gas gangrene, there is gas formation. So some of the spaces are occupied by gas, okay? So this gas formation is a typical feature of gas gangrene, okay? Again, you can see this is the gram stain done for muscle biopsy. You can see plenty of gram-positive rod-shaped anaerobic spore-forming bacilli. Okay, all these purple-colored structures are, gram-positive bacilli. So these gram-positive bacilli might be Clostridium perfringens, which is causing the infection. Okay. So this is a radiograph. This is the X-ray taken of a patient. Okay, the X-ray has been taken of, right shoulder. So you can see, can you see some blackish shadow over here? Here you can see, here you can see. These black shadows are not supposed to be there, okay? So all these black shadows are because of accumulation of gas, okay? If you see gas under the X-ray, it looks black, okay? There is a shadow, okay? So all this black shadow, this scattered black shadow over here, it's all because of accumulation of gas. So this is the histopathological demonstration of accumulation of gas. This is the radiological feature of gas, and this is the gram stain of muscle biopsy in which you can see gram-positive bacilli, which might be Clostridium perfringens. Okay, did you understand?
Yes,
Hello.
sir.
Yes, sir.
Yes. Can I move on to the next slide?
Yes, sir.
Okay. So what are the other diseases caused by Clostridium perfringens besides gas gangrene? It can also cause gangrenous appendicitis. Now you all know what is appendix, right? So this is what you call as appendix. It is present at the, some tip of this, ascending colon. This is the normal appendix. This is the inflamed appendix. In case of gangrenous appendix, this appendix might become black. It might become black just like the skin, okay? It can even cause biliary tract infection. Now this is the liver, this is the common hepatic duct, this is the cystic duct, this is the gallbladder. Infection of this, this parts is called as biliary infection. So it can even cause infection of the gallbladder, the common bile duct, cystic duct, okay? So it can cause infection of biliary tract also, okay? It can also cause brain abscess and meningitis. Now this is the CT scan of the patient. You can see this is the normal brain, and just look at this side of the brain. Okay, on the right side you can see a very ugly mass. Okay, a well circumscribed mass having, this periphery, the wall. So this might be the abscess. Okay, there is brain abscess. So when there is this kind of brain abscess in the patient, this patient is not going to become normal. Okay, there will be lots of clinical features like headache, photophobia, vomiting, loss of vision, okay, loss of hearing, personality changes, seizure, lots of things you might find in this patient with brain abscess, okay. Even it can cause meningitis also, okay. So our brain is covered by three layer, what you call as dura matter, arachnoid, pia matter. When there is inflammation of these three layers covering the brain, that is dura matter, arachnoid, and pia matter, it is called meningitis. It can also cause meningitis. And also it can cause panophthalmitis. Now look at the eye of this patient. The whole eye has been inflamed, okay? The whole eye. including the cornea and even the, the vitreous humor, aqueous humor, the whole eye has been involved. This is called panophthalmitis, means pan means whole thing, the whole eye has been involved. Even this can happen, okay? It can also cause thoracic infection. Sometimes the patient might have blunt trauma. The thorax might undergo trauma, some kind of stab injury, bullet injury, or road traffic accident. So in that condition, it may even cause thoracic infection of the thorax. Okay, and also it can cause infection of the urogenital system. That means it may cause infection of the kidney, ureter, bladder. Okay, it can cause inflammation of the urethra. It can even cause infection of the male and female genital organs also. Okay. So these are other infections caused by Clostridium perfringens in different parts of the body, okay, depending upon the site of infection. Okay, did you understand? Hello.
Yes, sir.
Okay, can I move on to the next slide?
Yes, sir.
Yes, sir.
So let's come to the diagnosis of gas gangrene. So clinical diagnosis, radiological diagnosis, and lab diagnosis. So if you are a good physician, if you want to become a good doctor, then we have to depend on different modalities of diagnosis. Like clinical diagnosis, you'll be doing by yourself, by looking at the patient, by examining the patient, by taking history of the patient. Okay, even if you are a good doctor and experienced doctor, then even by looking at the patient, you can diagnose. Okay, the lesion. Radiological diagnosis we do by doing CT scan, chest X-ray, MRI, ultrasonography. Lab diagnosis, we collect the sample from the patient, like the exudate, the necrotic tissue, etc., and we go for gram staining, culture, serology, PCR, etc. Okay, so these are the different modalities of diagnosis. So diagnosis of gas gangrene must be done by clinical examination, and lab only confirms the clinical diagnosis. See, the main diagnosis is by clinical diagnosis. When you look at the lesion of the patient, when you feel the lesion of the patient, then you can diagnose very easily. Lab diagnosis only for confirmation. Okay, the best diagnosis is clinical diagnosis in this case, and radiological diagnosis also might support the diagnosis. So for lab diagnosis, what sample we have to collect? We can collect the exudate. Okay, so you can see this is the exudate. Okay, you can see some kind of exudate present, serosanguinous exudate. Even this exudate might have plenty of bacteria. Okay, you can even collect the necrotic tissue. Even you may collect, the muscle fragment also. Exudate, necrotic tissue, muscle fragment can be collected. You may even, very rarely, you can collect the swab also. Okay, swab means just you have to rub this, this kind of cotton swab or Dacron or rayon swab on the surface of the wound and just do culture or gram staining. Curettage means in Nepali we can say khurkine, होइन, घाउलाई कोट्याउने अथवा खुर्केर sample लिने. Okay, curettage means घाउलाई खुर्किने अथवा कोट्याएर अलिकति दुख्छ होला तर you can do even by using certain anesthesia also, or you can even aspirate. Okay, you may aspirate the sample, the exudate from the deep lesion using the syringe also. So these are the different ways how you can collect the sample, and you can also collect the exudate, the muscle fragment, as well as the necrotic tissue for the diagnosis. Okay, did you understand?
Yes,
Hello.
Yes, sir.
Can I move on to the next slide?
Yes, sir.
Yes, sir.
Okay, so what are the diagnostic methods we do? We can go for direct microscopy. You can do gram stain, other different stain. Even you can do the biopsy, hematoxylin, eosin stain, or Gemsa stain. Then you must have to culture in anaerobic media. See, this is anaerobic bacteria. You cannot culture in aerobic media. You must have to make the media anaerobic, okay, suitable for anaerobic bacteria. That means the place where you grow this organism should be free from oxygen. If oxygen is present, then this organism is not going to grow, okay. Also, you can go for biochemical analysis. Last time I talked about, the Nagler reaction, Camp reaction, okay. Those biochemical reaction can be done for identification. Also, you can go for serological reaction. You can detect the antigen, antibody by different method, like you can do ELISA, okay. You can even do immunofluorescent assay, etc. Animal inoculation, this is not done routinely. Even you can use a lab animal, a guinea pig or mice, inoculate the patient's exudate. If the patient exudate has got Clostridium perfringens, the animal is also going to die after a few days. Okay. Then you can also go for molecular technique, that is the PCR. Now, all those unique genes which is present in the bacteria, which is not present in other bacteria, can be amplified and can be analyzed by using the molecular method. I think I've already taken lecture on PCR previously. Okay, what is the principle of PCR? What is how PCR is done? I don't have to tell you all this. Okay, So these are the different modalities of diagnosis. Can I move on to the next slide?
Yes, sir.
So direct microscopy, you can go for gram staining, in which you find gram-positive bacilli. You find a large gram-positive bacilli with or without spore that is strongly suggestive of Clostridium perfringens. So you can see here plenty of gram-positive bacilli having subterminal oval spore. So this might be Clostridium perfringens. You know, in case of Clostridium septicum and Clostridium novae, you may find subterminal spore, and you know the bacteria looks boat-shaped. The bacteria looks just like a boat. It has got boat shape or leaf shape, and it is pleomorphic in shape. It may be of different shapes and sizes. So this kind of boat shape or leaf shape bacilli is called as citron body. So you can see plenty of citron body in this. You can see the bacteria is taking a typical kind of shape, that is boat shape or leaf shape. So this is called as the citron body. It's also gram positive. Okay. Sometimes, you know, it is not necessary that gas gangrene should be caused only by Clostridium perfringens, septicum, and novae. There are other diseases which can also mimic gas gangrene. There are so many other diseases which looks very similar to gas gangrene. For example, anaerobic streptococcal myositis. When there is inflammation of the muscle by, you know, streptococcus, that is aerobic bacteria, in that condition also you might find the features are quite similar to gas gangrene. But when you do the gram stain from the sample, you are not going to find gram-positive bacilli, but you are going to find gram-positive cocci in chains. You are going to find plenty of pus cells with gram-positive cocci in chains. So this gram-positive cocci in chain is not Clostridium perfringens, but it may be Streptococcus pyogenes. This may be Streptococcus pyogenes. So even Streptococcus pyogenes can cause myositis, which can look quite similar to gas gangrene sometimes. Okay? Did you understand? Hello.
Yes, sir.
Yes. Can I move on to the next slide?
Yes, sir.
Yes, sir.
Okay. Now culture. Now all of you tell me, what is this? What you are seeing in this picture, Banada? What is this?
Robertsonian cooked meat broth.
Very good. Robertson's cooked meat media, right? This is Robertson's cooked meat media. This is the anaerobic culture media where we culture anaerobic organism. So samples are inoculated first on cooked meat broth. Okay. So you have to cook meat broth or RCM, or even you can culture in another media called as thioglycolate media or 1% agar. Okay. Here you must have to place the patient's exudate or the bit of necrotic tissue or the muscle fragment. And after culturing for some time, this is what you get. Okay, so when there is growth of bacteria, this liquid might be turbid. Even there can be changes in the color of the meat particle, indicating that the bacteria is growing within this. Okay. Now 4 tubes of RCM, you must have to take 4 tubes. Do not depend only on one tube. Take at least 4 tubes, and you must have to inoculate the sample and heat different tubes at different temperature. Okay. So 100 degrees Celsius for 5 minutes, 100 degrees Celsius for 10 minutes, 15 minutes, 20 minutes. Now, why we are heating at different time means the aerobic bacteria which we do not want is going to die, at 5 minutes, 10 minutes. Okay. Also, there may be certain heat resistant spores. Okay. Now, some heat resistant spore might get destroyed in 20 minutes. Okay. Might germinate in 20 minutes. Some heat resistant spore might germinate in 15 minutes. So you have to heat at 100 degrees Celsius at different times, 5, 10, 15, 20. The purpose of doing this is to kill the unwanted vegetative bacteria and to differentiate different heat-resistant spores of Clostridium perfringens. Okay, so do not depend only on one tube. You have to take about 4 different tubes, heating at 100 degrees Celsius for different amount of time. Okay. After culturing in this Robertson cooked meat media, you must have to subculture in neomycin blood agar. Okay. Now this is a neomycin blood agar. This is the blood agar in which neomycin has been kept, because neomycin is going to inhibit unwanted bacteria or aerobic bacteria. This Clostridium perfringens is highly motile. Since it is highly motile, it produces this kind of spreading growth. So this kind of rapidly spreading or moving growth is called as swarming growth. Okay. In order to prevent this kind of swarming growth, you must have to increase the concentration of agar in the media. Normally, only 1 to 2% agar is present in this media. If you increase the concentration of agar to 5 to 6%, then this kind of overgrowth or spreading growth is not found. So in order to stop this kind of over swarming growth or overgrowing, you must have to increase the concentration of agar to 5 to 6%. And also you must have to place a metronidazole disc and incubate anaerobically for 48 to 72 hours. Okay. So you can see here is the growth of Clostridium perfringens. Okay. It is showing some kind of hemolysis. This is the metronidazole disc. Are you finding any growth around the metronidazole disc here? Are you finding any growth around the disc?
No, sir.
No. Now, why we are not finding growth around the metronidazole disc means almost all anaerobic bacteria, okay, almost 100% of anaerobic bacteria, they will not grow in presence of metronidazole. Actually, metronidazole is the drug of choice for anaerobic infection. So if you place metronidazole disc and if you find there is no growth of organism around the disc, you will at least come to know that this bacteria is not aerobic, this bacteria is anaerobic. होइन metronidazole disc को वरिपरि यदि ग्रो भएन भने ल यो पक्कै पनि एनारोबिक ब्याक्टेरिया हो, यो एरोबिक ब्याक्टेरिया हुन सक्दैन भनेर तिमीले बुझ्छौ. त्यही कारणले that is why we must have to keep metronidazole disc, you know, in the media where we try to grow anaerobic bacteria. Okay, did you understand this slide? Hello.
Yes, sir. Yes, sir.
बुझ्या हो पक्का?
Sure, sir.
हजुर बुझ्यो?
Yes, sir.
बुझ्यो है? Can I move on to the next slide? Yes?
Yes, sir.
Okay. Now most strain they produce beta hemolytic colonies and few are non-hemolytic. Okay. The plate of serum egg yolk agar. You know, last time I talked about Nagler reaction, so I'm not going to repeat this again. So even you can do Nagler reaction, you know, for the demonstration of Clostridium perfringens toxin. The bacterial isolates are also identified by colony morphology, cultural characters, biochemical tests, and even reverse cam test. So I told you last time this reverse cam test, Christie, Atkin, Munz, Peterson test done for the detection of alpha toxin, right? I told you last time. Now these are the different anaerobic system. Okay. So some of the example of anaerobic system is this is Macintosh Fields jar. Okay. This is called as the gas pack system. Okay. So what they do in this Macintosh field jar is you can see two, you know, inlet and outlet. Okay. So this is inlet, this is outlet. So outlet is going to extract all oxygen present here. Okay. All the oxygen which is present here will be extracted from this outlet, and inlet you must have to inject, or you must have to put some non-oxygen gas in this. Okay, you might have to keep some gas which is not oxygen. Okay, even you can keep some kind of alumina coated palladium catalyst which is going to absorb or consume the oxygen. Now when this container becomes free from oxygen, okay, that means when this condition becomes anaerobic, then there is methylene blue kept at the side. This methylene blue, which is blue in color like this, will become colorless. So when there is absence of oxygen in this container, this methylene blue will become colorless from blue, indicating that now the condition is fulfilled. So this way you can create anaerobic chamber to culture anaerobic bacteria and keep the blood agar inside this neomycin blood agar with metronidazole disc. Okay, so this is the Macintosh fields jar which we'll be studying in practical also, don't worry. And this is the gas pack system. Okay, so this gas pack system also has got some kind of chemical which is going to consume oxygen. If there is oxygen, then this oxygen will combine with hydrogen to form water. So if oxygen is present here, all oxygen will be used and anaerobic condition will be maintained. So when anaerobic condition is maintained in this gas pack system, this methylene blue will become colorless. This methylene blue or the anaerobic indicator will become colorless. In this way, you can maintain the anaerobic environment to culture anaerobic organism. Also, you can use this kind of candle jar system. If you light a candle inside this, the candle is going to utilize all the oxygen present inside this jar and replace that with carbon dioxide. Hence, the anaerobic bacteria can grow. Anaerobic as well as carbon dioxide loving or capnophilic bacteria can grow inside this jar. So these are the different anaerobic system to maintain the anaerobic condition or to replace the oxygen for the proper growth of anaerobic bacteria. Okay, did you understand? बुझ्यो?
Yes, sir.
बुझ्यो है anaerobic condition. तिमीहरुको anaerobic system को लेक्चर भएको छैन होला है? भयो?
भइसक्यो सर
सुरेन्द्र सरले पढाएको हो हेलो बुझ्यो हेलो
यस सर
okay can I move on to the next slide
यस सर
okay now also you can go for animal pathogenicity okay so 0.1 ml of 24 hours growth in cooked meat broth is injected into healthy guinea pig okay so this is guinea pig you must have to inject 24-hour growth of the bacteria inside this guinea pig intramuscular the animal will die within 24 hours okay the animal dies within 24 hours and if you cut open this animal you will find presence of bacteria inside the viscera inside the body inside the heart or spleen of this animal actually you have to take two animals one animal is protected with antitoxin okay prior the test antiserum the animal which has been protected with the antitoxin will survive that is lucky animal but the animal which has not been given antitoxin injection is going to die so after that animal dies you must have to do necropsy of that animal and inside the animal organ you can find plenty of the bacteria so you can see this part of that animal organ has undergone gangrene see it has become black that means it has gone gangrenous infection okay there is gas gangrene of this part okay of this animal so in this way you can demonstrate the effect of that bacteria in animal also okay but this is not done routinely there are some issues regarding animal rights also okay so this has to be done only after taking special permission okay can I move on to the next point
यस सर
बुझ्यो है यो so you can also do ELISA for detection of toxin okay now this is what you call as sandwich ELISA okay sandwich ELISA why it is called because the antigen is being captured between two antibody okay so this is the toxin this target protein is the toxin which we want to detect this is the ELISA plate which looks something like this in this plate there are some micro titer wells in that well at the base there is capture antibody coated okay it is fixed capture antibody when you add the patient sample having the antigen that antigen will be trapped will be caught by this antibody okay again you have to add another antibody which will act as detection antibody so now you have got two different types of antibody in between you have got antigen again you have to add another antibody which is the animal antibody so this antibody is antibody to human antibody that means it is the anti-antibody okay anti-antibody means animal antibody antibody to human antibody but this antibody is labeled with some kind of enzyme HRP that is horseradish peroxidase okay horseradish peroxidase if you add substrate TMB that is tetramethylbenzidine will react with horseradish peroxidase to produce this blue color so if you find blue color that indicates presence of this antigen or the toxin if this antigen is not present in the patient's serum then this antibody cannot bind with this antibody and even this antibody will not bind with this antibody you will not find any blue color so absence of blue color will indicate negative test presence of blue color will indicate positive test so this sandwich ELISA test can be done for the detection of antigen or the toxin did you understand all of you बुझ्यो
सर
बुझ्यो है तिमीलाई एलाइसा त पढाएको होला नि हैन उसमा आई थिंक इम्युनोलोजीमा एलाइसा पढाइसक्यो होला है पढाएको हो
हो सर
okay so also you can go for PCR for detection of toxin okay so what is PCR Bhanada what is PCR खै त बोल न can someone speak please what is PCR खै hello
DNA amplification.
what is the full form of PCR
Polymerase
Polymerase
oh भनौं त अनि भन polymerase chain reaction okay now this PCR is going to detect the gene which is responsible for producing the toxin the unique gene okay so what can be the target gene what can be the primer alpha toxin beta toxin epsilon toxin I talked about iota toxin right? So these unique sequence you can see GTP GA okay guanine thiamine guanine adenine so these unique gene sequence which is responsible for producing this toxin can be detected, can be analyzed, can be amplified by PCR. So PCR is very accurate, very sensitive and specific method for diagnosis, but it is quite expensive. We need a special equipment, and the person who is doing PCR should be really trained, really well trained, and we need a very good room free from any contamination. Okay, so even we can do PCR for gene detection, gene for toxin production detection. Okay, did you understand? Hello?
Yes, sir.
Yes. भनेपछि you can go for staining, you can go for culture, you can go for serological test, you can go for animal pathogenicity, you can go for biochemical test, you can go for PCR. These are the different methods of diagnosis of a gas gangrene or Clostridium perfringens infection. Can I move on to the next slide? Yes? Hello?
Yes, sir.
Okay, so treatment and prevention. Surgery is the most important prophylactic and therapeutic measure of gas gangrene. हैन, सर्जरी गर्नुपर्छ, अपरेसन गर्नुपर्छ है. अब हात कुहिएको हो भने हात काटेर फाल्नु पर्यो, खुट्टामा इन्फेक्सन भएको हो भने खुट्टे काटेर कहिलेकै फाल्नु पर्ने हुन सक्छ है, एम्पुटेसन गर्नुपर्ने. All damaged tissue, okay, all damaged tissues should be removed promptly and the wounds irrigated to remove blood clot, necrotic tissue and foreign material. जति पनि कुहिएको सडेको टिस्युहरु छ त्यो सबै हटाउनु पर्यो, किनभने त्यो फैल्यो भने मान्छे मर्न सक्छ है. होल थिङ यु हेभ टु रिमुभ. लिम्ब एम्पुटेसन मे हेभ टु बी डन, हैन. लिम्ब भनेको हात अथवा खुट्टा काट्न पर्ने हुन सक्छ. लिम्ब एम्पुटेसन मे हेभ टु बी डन. In established gas gangrene, uncompromising excision of all parts are maybe life-saving. So in order to save the life of the patient, a certain parts should be sacrificed. You must have to remove certain part of the body. You know, you can even go for hyperbaric oxygen therapy. भनेको, if you do high pressure oxygen therapy, that can kill the bacteria, since the bacteria cannot survive in presence of oxygen, and since oxygen can provide, beneficial effect to the patient, you can do high pressure or hyperbaric oxygen therapy. So hyperbaric oxygen therapy is done in such kind of chamber. Okay, this is the hyperbaric chamber. The patient is given high concentration of oxygen in order to provide, some beneficial effect as well as to make the environment unfavorable for the bacteria. So this is the hyperbaric oxygen therapy. Okay, did you understand?
Yes, sir.
Hello?
Yes, sir.
Sir. Yes, sir.
Can I move on to the next slide?
Yes, sir.
Yes, sir.
Now, also you can give antibiotics like metronidazole. Okay, I told you metronidazole is the drug of choice for anaerobic bacteria. You can give metronidazole intravenous before surgery and repeat it 8 hourly for 24 hours. Surgery गर्न अगाडि पनि सर्जरी गरिसकेपछि पनि you must have to give this anti-anaerobic drug or metronidazole. Then for the other bacteria which can cause contamination, you also have to give broad spectrum antibiotics to cover the aerobic infection. So there can be aerobic as well as anaerobic infection combined. So in order to kill the aerobic bacteria also, you have to give broad spectrum antibiotic. So you can give, beside metronidazole, other drugs like you can give gentamicin, amoxicillin, ampicillin, sulbactam combined either with clindamycin or metronidazole. So if you give these drugs, it is going to cover the gram-positive as well as gram-negative aerobic bacteria, which can also cause cross-infection along with Clostridium perfringens. Okay. You also have to immunize the patient. Okay. You must have to give anti-gas gangrene serum. That means you must have to give antibody to the patient. That antibody can, neutralize the effect of toxin. Okay. Now for immediate protection of the patient, in order to save the life of the patient and decrease the action of toxin, you must have to give anti-serum. So anti-serum contains antibodies to toxin of Clostridium perfringens. It is a polyvalent antitoxin in a dose of 10,000 international unit Clostridium perfringens, 10,000 international unit Clostridium novae, and 5,000 international unit Clostridium septicum. So this antitoxin or anti-gas gangrene serum is going to cover for all these three bacteria, all these three kinds of bacteria, which can be harmful to the patient. Okay, so if you give this anti-gas gangrene serum, then this can be life-saving. It is given intramuscular to the patient, which can save the life of the patient because this antitoxin can neutralize the toxin, right? Okay, did you understand?
Yes, sir.
Yes, sir. Okay, so this finishes your Clostridium perfringens and gas gangrene. Okay. So should I start another topic or next time?
Next time.
Next time. आज यति गर्ने कि अलिकति सुरु गरौँ अर्को टपिक?
No sir, next time.
हजुर? तिम्रो जे भन्छौ के गर्छु भन त?
Next time गरौँ सर. हामी पढेर आउँछौँ. Next time.
अब सुन त के रे मलाई इमेल मेसेज गर है तिम्रो
हजुर मैले भाइबरमा गरिसकेँ
है. ओके अनि म तिमीलाई यो आज र यो अस्ति पढाएको यो लेक्चरलाई म यसको पीडीएफ फर्मेट तिमीलाई पठाइदिन्छु है. हुन्छ? अनि तिमीहरू चाहिँ कुनै फ्याकल्टीसँग अरु कुनै कलेजको साथीहरुसँग अरु बाहिर कलेजकोहरुसँग शेयर नगर प्लिज. ओके
Sure,
intellectual property please I don't like it to share with other except you all. तिमीहरुलाई मात्रै अधिकार छ है, तिमीहरुको लागि मात्रै बनाऊ है त. ओके
Thank you sir. Thank you
please
sir.
make sure about that है. त्यसको तिमीहरुले चाहिँ एउटा प्रतिज्ञा गर्नु पर्यो है. ओके
Sure sir.
okay thank you all of you. Can I